The title of this lecture is Migration. Migration is the third evolutionary force of evolutionary change, that is. It is defined as the movement of genes from one population to another, and it is also known as gene flow, the movement of genes from one population to another. The movement of these red ladybugs into the white ladybug population, the movement of the white ladybugs into the red ladybug population. If all the individuals in the classroom population do not choose their mates from within the group, significant change in allele frequencies are possible. For example, if 4 people who had A blood moved out and 4 people who had O blood moved into the population, the allele frequencies would be altered. If a change in the allele frequencies occurs, then we say that evolution has occurred. And this is due to the force of migration. Gene flow, like mutation, can introduce new genes into a population. However, in this case, the genes are only new to the population and they are not new to the species. Gene flow decreases the differences between populations, making populations more genetically similar, more homogeneous. Let's take an example. If we have population 1, which has all dominant alleles, so the frequency of the dominant alleles is 1, the frequency of the recessive alleles is 0. In population 2, we have no dominant alleles, so the frequency of the dominant alleles is 0, and the frequency of the recessive alleles is 1. Now, let's hypothesize that if 10% of population 1 moves into population 2 and 10% of population 2 moves into population 1, this constitutes gene flow. What effect will it have on these populations? Well, after gene flow has occurred, population 1 is going to be 90% dominant alleles and 10% recessive alleles. Population 2 is now going to have 10% dominant alleles and 90% recessive alleles. The allele frequencies have become more similar. If this same rate of 10% of gene flow continues, the similarity between population 1 and population 2 is going to increase rapidly. After 20 generations, the two populations will be nearly identical. Humans are very mobile creatures, especially relative to other biological organisms. Human populations show a tremendous degree of variation in their degree of migration. Some humans choose their mates from relatively close to home. Others move away far from home and choose their mates from far away. Now, humans all live and work within a small area, and some are more mobile than others. The gene flow between these human populations, the ones that choose a mate from close by and the ones that choose a mate from far away, acts to reduce those differences between the groups. It makes human populations more homogeneous. Studies of migration and gene flow look at the distance between birthplaces or premarital residence of married couples. And it has shown that the amount of gene flow depends on environmental and cultural factors. Geographic distance, for example, is a major determinant of gene flow. The farther apart two populations are, the less likely that they will exchange mates. For example, if you were born in New York City and your spouse was born in Chicago, the distance between your birthplaces would be something like 800 miles. If you were born in the same neighborhood and you chose your spouse from that neighborhood, the marital distance would be 0. Most marriages actually occur within just a few kilometers, and the number of marriages is known to decrease as distance between populations increases. So the farther apart you are, the less likely you are to choose a mate from that distance. The closer you are, the more likely you are to choose a mate from close by. The majority of gene flow in human populations occurs in local areas. Smaller proportion of gene flow occurs farther away. Let's look at an example. Okay? There was a study in Leominster, Massachusetts, and they explored marriage records from 1800 to 1849, so a 50-year time period. And they recorded 602 marriages during this 50-year period. 49.2, nearly 50%, were between people that were native to Leominster. So 50% of the population chose their mates from the town in which they were born. 24% lived 5 to 10 kilometers apart, 16% lived 10 to 20 kilometers apart, and 10% lived over 20 kilometers apart. So the majority of them were very, very close, and only a small number of them were marriages from a distance away. The point that you need to understand is long-range migration keeps populations from diverging too much from the rest of the species. So all you need is a little bit of gene flow from the edges of your population to keep it homogeneous with the rest of the population. So let's take another example about gene flow. Now, let's say there are 100 people in a hybrid population. And we're going to create this hybrid population from population 1 and population 2 that we talked about before. Population 1, the frequency of the dominant allele is 1, and the frequency of the recessive allele is 0. Let's say there are 90 people in population 1. In population 2, they have no dominant alleles, and the frequency of the recessive alleles is 1. And there are 10 individuals in population 2. The hybrid population is going to be comprised of population 1 plus population 2. So we're going to combine population 1 and population 2 for 100 individuals and create a hybrid population. The question we now want to ask is, how can we determine the allele frequencies of this hybrid population? We can do this in two ways. First, we determine the allele frequencies in population 1 and the allele frequencies in population 2. After we determine the allele frequencies in both populations, we now calculate the relative contributions of population 1 and population 2 to the hybrid population. So, at a single locus with two alleles, so we have a single spot on the chromosome and there are two alleles, the dominant and the recessive. In population 1 will equal P1, and that constitutes the frequency of the dominant allele in population 1. And we already said in the previous example, or in the previous slide, that the frequency of the dominant allele in population 1 is 1, that all the individuals in population 1 have the dominant allele. We also said that the frequency of the recessive allele in population 1 is 0. So we have Q1 will equal 0, P1 will equal 1. P2 and Q2 represent population 2. P2 is the frequency of the dominant allele in population 2. Q2 is the frequency of the recessive allele in population 2. We already said in the previous slide that the frequency of the dominant allele in population 2 is 0, and the frequency of the recessive allele in population 2 is 1. So P2 will equal 0, and Q2 will equal 1. That's the allele frequencies. Now we need to calculate the relative contribution of population 1 and population 2 to the hybrid. M1 and M2 represent the relative contribution of population 1 and population 2 to the hybrid. M1 is the percent of the hybrid population that comes from population 1. M2 is the percent of hybrid population that comes from population 2. If you remember from a couple slides back, we said there were 90 individuals in population 1, and we said 10 individuals in population 2. So 90% of population 1, of the hybrid population, 90% of the hybrid population is coming from population 1, 10% of the hybrid population will be coming from population 2. The thing to keep in mind is M1 and M2 are always going to equal 1. So the next thing that I want to mention is that we now calculate hybrid gene frequencies by taking what's called the weighted average of the gene frequencies in the contributing population. We basically calculate the weighted average of the gene frequencies in population 1 and population 2. This is how we do it. This formula. P is the frequency of the dominant allele, and H represents hybrid, so that we're looking at calculating the frequency of the dominant allele in the hybrid population. And we say that it equals M1 times P1 plus M2 times P2. If you'll recall from the previous slide, M1 equals the percent of the hybrid that comes from population 1. In this example, it was 90 individuals out of the total 100 individuals in both populations. So that equals 90 divided by 100, or 0.9. So we put 0.9 in right down here. P1 equals the frequency of the dominant allele in population 1. Remember, we said the frequency of the dominant allele in population 1 was 1. So now we put a 1 down here. M2 is the percent of the hybrid population from population 2. We said there are 10 individuals in population 2 out of the total of 100 individuals in our hybrid population. That equals 10 divided by 100, or 0.1. So we put 0.1 down here. And P2 equals the frequency of the dominant allele in population 2. And we have no dominant alleles in population 2, so we put in a 0. 0.9 times 1 equals 0.9. Plus 1.1 times 0, which equals 0. So we add this up, and it equals 0.9. So the frequency of the dominant allele in the hybrid population equals 0.9. Okay, so let's take an actual example. What are the allele frequencies in the hybrid population? We need to calculate first the relative allele frequencies of each population and calculate the relative contributions of each population. In this example, in population 1, we have 1,200 individuals, and in population 2, we have 300. So the hybrid population is going to be 1,500 individuals. 0.25 is the frequency of the homozygous dominant genotype in population 1. 0.5 is the frequency of the heterozygous genotype in population 1, and 0.25 is the frequency of the homozygous recessive genotype in population 1. In population 2, the frequency of the homozygous dominant is 0.09. The frequency of the heterozygous genotype in population 2 is 0.42, and the frequency of the homozygous recessive genotype in population 2 is 0.49. Once again, all of these in population 1 and 2 equal 1. So the first thing we need to do is calculate our allele frequencies. In population 1, the allele frequencies, the frequency of the dominant allele A equals 0.25 plus 1/2 divided by 2. This is 0.25 is the frequency of the homozygous dominant plus 1/2 of the frequency of the heterozygous genotype. The heterozygous genotype was 0.5 in population 1. We divided by 2 because this heterozygous genotype has both A alleles and B alleles. And we get the frequency of the dominant allele, A, in population 1 is 0.5. In the frequency of the recessive allele in population 1 is 0.25 plus half of 0.5 equals 0.5 equals Q1. In population 2, A, the frequency of the dominant allele in the homozygous dominant genotype is 0.9 plus the frequency of the heterozygote is 0.42. We divide that in half because it has both alleles, and we get the frequency of the dominant allele is 0.3. The frequency of the recessive allele is 0.49. This is the frequency of the homozygous recessive plus 1/2 of the frequency of the heterozygous genotype equals 0.7. So we have all of our allele frequencies. These are the allele frequencies for population 1. These are our allele frequencies for population 2. Now we have to calculate M, the relative contribution. Population 1 has a total of 1,200 individuals, and the hybrid population has 1,500. So 1,200 divided by 1,500 is 0.8, which equals M1. Population 2 has 300 individuals. We divide it by the total number of individuals in the hybrid population, and that equals 0.2, or M2. So now we have the frequency of the alleles in population 1, the frequency of the alleles in, the frequency of the alleles in population 2, and the relative contribution of population 1 and the relative contribution of population 2. So now we have to use that formula. The pH equals M1 times P1 plus M2 times P2. This is the frequency of the dominant allele in the hybrid population. P of H, the frequency of the dominant allele in the hybrid population. We take the numbers from the previous slide. P of H, this is the frequency of the relative contribution of population 1. This is the relative contribution of population 2. P1 equals 0.5, P2 equals 0.3. We calculate it out. 0.8 times 0.5 is 0.4, plus 0.2 times 0.3 is 0.06. So we have the total for the hybrid of the frequency of the dominant allele in population 1 as 0.46. The frequency of the B allele in the hybrid is done in an analogous manner. Q of H equals M1 times Q1 plus M2 times Q2. So we're figuring out the frequency of the recessive allele in the hybrid population. The relative contributions of population 1 and 2 are the same as in the previous formula. What's changing is the Q's. Q1 equals 0.5, Q2 equals 0.7. And the frequency of the recessive allele in the hybrid population is 0.54. Notice 0.46 and 0.54 equal 1, so we know we have done our mathematics correctly. I want to point out that it's relatively unusual for an anthropologist to be asked to predict frequencies in a potential hybrid population. We're never really said, combine this population with this population. We're not told to do that. What we are told to do, or asked to do rather, is try to figure out which populations arose from a mixture of what population. And anthropologists are usually asked to reconstruct how populations have mixed together. Okay, let me give you a specific example. Excuse me. We know, for example, that American blacks are descended from slaves that originated from West Africa, predominantly from West Africa. We also know that American blacks have interbred with the U.S. white population. Some might call it the European U.S. white population. The question is, can we determine what percent of the genes that are in American blacks are actually from their African roots or from their admixture with the European U.S. white populations? We can actually solve this question by solving the formula for M, for relative contribution, instead of proportion of dominant alleles in the hybrid population. So here we're solving for M2, the proportion of the genes that came from population 2, instead of solving for the proportion of the recessive alleles in the hybrid population. You don't need to know how we solve for M versus Q. You do need to know these formulas though. So M2 equals QH minus Q2 divided by Q1 minus Q2. The relative contribution from population 1, M1, equals pH, proportion of the dominant allele in the hybrid, minus the proportion of the dominant allele in population 2, divided by proportion of the dominant alleles in population 1 minus the proportion of dominant alleles in population 2. These formulas will be available during the exam. Okay, you just need to know how to put the data in them. When solving for M, for solving for the relative contribution, it's very important to choose an allele for which you think the ancestral populations will differ. One allele that African blacks and European white populations are known to differ is the Rh allele. The Rh blood group is named for the rhesus monkey, in which it was first discovered, and it consists of people who can make the Rh antigen, and they're generally referred to as Rh positive, and people who cannot make the Rh antigen, Rh negative. Some of you may know that your blood type is Rh positive, or is A positive or B positive or AB positive. When you're positive with A, they're actually combining two different blood types, your actual blood type and the rhesus antigen. So the plus or minus in your blood type refers to whether your body can or cannot make the rhesus antigen. The Rh blood group is of major concern because it leads to immunological incompatibility between moms and their fetuses. This is known as a condition known as the hemolytic disease of the newborn, HDN, and it primarily occurs when the mother is Rh negative, so her body cannot produce the Rh antigen, and the fetus's body is producing the Rh positive antigen. What happens is blood from the fetus, the Rh positive blood, enters maternal circulation through the placenta because it's not a perfect transition, and the antibodies against the Rh positive will be made by the mother. So the mother's body tries to produce antigens to get rid of the Rh positive antigen, and it can cause fetal death. It can also end up causing maternal death as well. So it's a very, very dangerous condition. And when I say that there are differences between the groups, we note that in West Africa, the frequency of Rh positive, we might call this Q1, is 0.55. So about 55% of the population in West Africa have Rh positive alleles. In the U.S. white population, ancestral from Europe, the frequency of Rh positive, we might call this Q2, is 0.03. So 3% of the U.S. white population is Rh positive, and the frequency in West Africa for Rh positive is 55%. So there's a big difference. Now let's look at American blacks. What's the frequency of Rh positive for them? They are going to be considered the hybrid population because they're a mixture of West African ancestry and admixture or interbreeding with the U.S. white population. And the frequency of the Rh positive allele, the proportion of the recessive allele in the hybrid population, Q of H, is 0.45, or 45% of them have Rh positive. So we're going to say it's a mixture of the two populations. The question is, can we determine what percent of the Rh positive alleles in African black, let me restate that. Can we determine what percent of Rh positive alleles in U.S. blacks and American blacks population that are from their African ancestry versus admixture from European populations? And the answer is yes, we can, by solving for M. M1 is the frequency of equals Q of H, the frequency of the recessive allele in the hybrid, minus Q2, the frequency of the recessive allele in population 2, divided by Q1, frequency of the recessive allele in population 1, and Q2, the frequency of the recessive allele in population 2. So let's take those numbers from the previous slide. We said that the frequency, the hybrid population, the mixed population of American blacks for Rh was 0.45. We said that in population 2, the U.S. white population, the frequency of Rh positive was 0.03. We said that the frequency in population 1, the original West African population for Rh, was 0.55. and we then subtract the frequency of the U.S. white population, RH, which is 0.03, and we get the frequency of M1 being 0.8, and the frequency of M2 equals 0.2. So we can say that 80% of U.S. American blacks, their RH alleles are from their African ancestry. 20% of the RH alleles in mixture are due to admixture with the U.S. white European populations. Isn't that kind of cool? I think so. So we can determine how much of the genes have mixed and how much haven't exclusively by solving for M, the relative contribution. Okay, so once again, 80% of the Rh positive alleles in American blacks are from the ancestral West African population, and 20% of the white population contributed their genes to the American black population, which is why we have 20% admixture.